Good morning.
Good morning. All right, so where are we? Let's get my... That's pretty funny. Hello, Connie. Okay, where are we at? All right, so sorry about Monday. Let's see. Okay, so essentially, going off of the environments that we've looked at on Europa and Enceladus especially, but also Titan, which is, pretty chilly. Sorry. One of the key attributes is cold tolerance, obviously, or ability to live at low temperatures, and temperatures are really low on those places, at least in parts of them. And the other big thing is halo tolerance, because basically the argument for liquid oceans on Europa, or under the surface of Europa and Enceladus, are that those oceans are salty, right? That's pretty much partly how they are detected, right? So the subsurface ocean on Europa was detected in part through the plumes affecting the magnetic field of Jupiter, right? And they'd only be able to do that if they were if they contained ions like sodium and chloride, presumably, or some other kind of salt ions. And in Enceladus, the argument is similar, but more based on temperature, right? So essentially, they're cold, salty environments, right? That's the deal. And so really, we want to be looking at where we find, cold, salty environments on Earth. And the discovery of those oceans on those two moons really did boost the study of halophiles considerably, and in terms of how they work. People were studying them before and just out of curiosity, but now it's a much more directed research. Also, what I want to kind of reinforce as well is that an awful lot of these adaptations are crossover. There's a lot of crosstalk between them. And so, hello, dog. My teenage dog has just come downstairs. There's a lot of crosstalk between these different stresses. Hello. Here she is. It's kind of early for you, dog. She probably needs a pee or something. I might have to get up and let her out in a second. What's going on in chat? Is this admiration of my dog? Yes. She is super cute. She's a scaredy pup. She's scared of everything. Scared of the laundry basket, scared of the watering can. She is 2 and a bit. We got her the month after we came to San Antonio in July 2017. Oh, she's not that big. She's kind of chunky. We call her a potato because she looks kind of potato-like. Anyway, before we get too distracted on Doggo. And her name's Luna because she's completely nuts. She's a lunatic, aren't you? She loves her people as well, which is good, obviously. Completely useless guard dog, though. She's just too interested in strangers. She thinks they're coming to say hi to her. Oh, that's cute. Luna's a popular name for dogs, it seems. Some other people we found have dogs called Luna too, perhaps because they're crazy. Anyway, for before I get too distracted on dog stuff. Stop that. Let's stick that over here then. All right, so there's an awful lot of overlap, right? So typically if you are able to resist osmotic shocks, for example, then almost certainly you're capable to resist high salt concentrations. So also that kind of bleeds into desiccation resistance. It's the same kind of principle essentially. And also, obviously, resistance to cold is very similar in many ways. In fact, there's an awful lot of overlap between that and resistance to high pressure. So, and also radiation resistance, right, which is kind of crazy. So one of the high salt tolerant organisms, where is it? This one here. Halobacterium salinarum. So this is an archaeon. It's not actually a bacterium, which is kind of confusing. This is also extraordinarily radiation resistant, almost as resistant as Deinococcus radiodurans, right? So there's a lot of overlap. Typically, if an organism can tolerate one environmental stressor, then it is able to tolerate others as well. Which kind of gives you a sense of how those traits evolved, right? It's not just, and we talked a bit about this with radiation resistance, it's not just resistance to one particular thing, it's that it also confers a selected advantage to other stresses too. Let's get this up here. I'm going to stick this. No, there we go. All right, that'll do. Also, there are such things as halophilic sheep. Did you know that? I didn't know that. I think that's nuts. See if I can stress out Paul a little bit. I'm going to find there. There you go. There they are. These are sheep that are able to grow, live off of kelp and seaweed that's washed up onto a seashore. Don't know why. They're off a island north of Scotland called the Orkney Islands, which are one of a island chain, kind of way up in the North Sea, northern North Sea, North Atlantic. Anyway, they can live off of a high salt diet essentially, which is kind of crazy. Never knew that such things existed before. Cool environment though. I actually met one day a woman and her family who were on a train who were traveling back from a wedding. So it took them 3 days to get from their house on the Orkneys to London, which is kind of crazy. And they actually lived on an island which was kind of saddle shaped. And in winter, when there was these big storms, they used to have waves that would hit one side of the island, and they'd actually the water go over their house into the sea on the other side of the island, which is kind of crazy. Anyway, that's a bit of a tangent, which is no big surprise, right? Anyway, so typically high salt environments. So there's obviously a range, right? Sea water is about 3.5% salt, give or take, weight by volume, and obviously these organisms vary in their tolerance to salt, and so what are called extreme halophiles, which would be these algae here, Dunalilia, and also Artemia sea monkeys are pretty salt tolerant too, but Halobacterium, they can live on salt that is actually saturated. So a saturated salt solution, which is around between 30 and 35%. And so the saltiest places on Earth typically they max out at that. So one is Lake Assal, which is in the Great Rift Valley. Actually, you'll tend to find there's a bit of a theme here. Salty places are typically in geological rifts, right? So kind of essentially valleys that are lower than than sea level, right? And so the Jordan Rift Valley is another one, which you probably all heard of. The Dead Sea. Oh no. I don't know who that is. Probably some scam call. That's good. Always nice to have a phone call that I don't have to answer. Yeah, so the Dead Sea is another one. That's one of the saltiest places. That's about 34% salt concentration. And the Dead Sea, the shore is about 1,400 feet below sea level. So that gives you an idea of part, and that's same for the Great Rift Valley. Is also part of the reason why they're so salty. They're very dry places, and they're typically not fed by many rivers. So Lake Assal is one of those. That's in Djibouti in Africa. So you see it's near Somalia and Yemen. The other place which we've also seen before is dry valleys in Antarctica. Right, so this is Don Juan Pond. Don't know why it's called that. I think there's something in Wikipedia that tells you why. And that's about that deep, give or take. And it never freezes, right? So it's so salty. It's about 44% salt concentration that it would have to be below minus 60 or minus 65 degrees Celsius for it to freeze. And it's only about 4 inches deep, barely enough to paddle in. Nothing living there. Same as the rest of the valley, actually. It's a complete dead zone. And this also is a pond which is not, it has no outlet, right? And that's quite common as well. Same with the Dead Sea too. It doesn't have any outlets. It just has a single inlet, which is the Jordan River. Anyway, so we've got a couple of things to talk about today. We've got halo adaptations to high salt, and we've also got adaptations to low temperature, and there's some fun stuff to talk about down here, including let's kind of wander around a little bit. Has anybody ever seen Fat Bear Week? If not, you're in for a treat. You might have seen this, Paul. You're a fan of the esoteric like I am.
I can't say I know what this is. I'm sorry to let you down.
Super cool. That's all right. No judgment here. This is a place up in British Columbia, in Canada. And they have bear cams, right? You can see one here. You can play that later. There's just some bears fighting over salmon. And basically, they follow these bears, or you can yourself follow these bears. It is body positivity. That's right. Yeah. You can follow them over the course of the year, and basically they start off super skinny because they've just come out of hibernation, and then basically you vote on which you think is the chunkiest bear, basically. And so it really is a go fat bears kind of competition, and the fattest and chunkiest bear wins. I don't think they get a prize, you probably wouldn't want to go near them to give them to it unless you had like loads of salmon to throw at them or something. But anyway, this is Fat Bear Week. It's in Katmai National Park, and it's super cool because you can just, if you're super bored or trying to avoid studying or something, you can watch these bear cams and just watch them catch a salmon in their teeth and scratch their asses and do what bears do. But anyway, there you go. So I thought I'd share that with you because I always think it's super cool. Obviously, it's gone for this year, but it'll be happening again in 2020 and this autumn. So keep an eye open for Fat Bear Week. It's lots of fun. Anyway, bouncing back to halophiles. So essentially, there's two kind of real adaptations, right? And this also semi-defines how tolerant they are of salt, right? So the moderately tolerant and the kind of the in-between tolerant ones, they tend to use more osmoprotectants, right? And you'll actually see the same thing down, where are we at? Here, right? So the same kind of ideas, right? Trehalose, sugars, amino acids, things like that, that protect against cold, also protect against high salt concentrations, right? So these are often used, these haloprotectants, I guess you could call them, are often used to protect against transitory spikes in salt concentration, right? And also to adapt to less extreme salt concentrations. Now, the other group, the extreme halophiles, they actually use light-powered sodium pumps, right? Not directly. They use something called bacteriorhodopsin, which I know Angela's heard of. If you studied micro, you might remember it from there. I think that's where she learned it. But essentially, what it is, it's a protein that is uses energy of a photon to change its conformation and pump a proton across the bacterial membrane out of the cell. And so this is a characteristic of all extreme halophiles, is that they actively pump sodium ions out of their cells. And that's essentially how they control their internal osmolarity, right? Along with these haloprotectants as well. And so by pumping these protons out, they use basically a proton-powered sodium pump to pump sodium out of the cell, and that's typically balanced out by potassium coming in. And so they're able to create extraordinarily high salt concentrations outside of their cells, up to like 5 or 6 molar sodium concentrations. And you can actually see it here, right? These bacteria, no, not bacteria, sorry, archaea, they're actually growing on salt crystals, right? And I think, I'm not 100% sure, I think these salt crystals are in part forming because of their salt pumps, that they're pumping these sodium ions out, and that's forming more salt crystals around them. This bacteriorhodopsin, there's also halorhodopsin as well, which is a chloride, light power chloride pump, which also helps balance charge. This bacteriorhodopsin is also a pigment, right? So it's kind of purplish color. And so if you look at these pictures here, which are super pretty, these are salt pans, right? So places where they actually mine salt. You'll see that that purplish color, that is generally because of bacteriorhodopsin. And where was it? Yeah, I think it was the Dead Sea. They had like a lot of rain or something, like a real rare event where it rained heavily and the salt concentration dropped. And when it did, they actually had a bloom, an algal bloom, right? So the sea turned green, which was largely due to this algae here, Dunalilia. And then it turned kind of like a purplish color, purplish red color, because of these microbes that were also feeding off of the algae and other nutrients that they produce. Oh, doggo. Luna's happy to see everybody else come down from upstairs. Anyway, so yeah, you actually, when you see when the salt concentration drops, you get these huge algal blooms, because basically they've been hanging on and more or less surviving at that high salt concentration, and then when the salt concentration drops a little bit, they're able to go full out. And also these carotenoids, right, which basically make this algae greenish orange color, also responsible for these greeny orangey yellow colors, and they're typically UV protectants, right? They also help with osmolarity as well, and membrane structure, but they also absorb UV radiation. So it's again another aspect of that kind of crosstalk between different environmental stresses, right? So these are improve the radiation resistance of these organisms, because typically these things grow places that are, you know, sunny. They grow in, you know, shallow films. They're not very far from the surface, right? So they're exposed to a lot of UV radiation as well. Anybody got any questions about halophiles before we move on? It's kind of a bit of a wild ride through them, to be honest. Nope. All right, so now we get on to psychrophiles, which is a cool word for liking the cold. Could also say British people. Actually, we don't like the cold. We complain about it all the time. We like our 2 weeks of sunshine that comes every summer. Anyway, so there's a lot of argument and dispute about psychrophiles, because really it's how you define tolerating the cold, right? There's an awful lot of things like bears that can tolerate the cold, but they don't they don't reproduce and grow during Arctic winters, right? They go to sleep, they hibernate. Same can be said for some of the funky fish that you find in the poles. Some of them don't actually grow and reproduce in the cold, they freeze, which is super cool, right? It's not to be trivialized, obviously. But it's kind of like a different approach, right? Whereas there are some other organisms, such as wingless midges, which sounds fabulous to me. I hate midges. They're like small, annoying mosquitoes, and there's loads of them, particularly in Scotland. Lichens and these various, I can't quite see them very well. Let's move this over. There you go. These bacteria actually grow and reproduce at low temperatures. This lichen, which we saw on the space station, right? It can quite happily grow and photosynthesize at -24 Celsius, whatever that is in Fahrenheit. I've absolutely no idea. Wingless midges can also do the same. So there are lots of different approaches to cold, right? One of them is to adapt to it and grow and reproduce. You tend to see that more in simpler organisms, right? Insects and below. Fish as well, to some extent. Other organisms, you'll see more behavioral and physiological adaptations which allow them to survive being very cold and then continue growing and reproducing when it's not quite as cold. So anybody heard of this species before?
The frog? No, I
Yeah.
literally just Googled it and there is no results on it. I was so disappointed. You broke my heart.
Hey, don't believe everything your professor tells you, right?
Like, I was like, well, there's no way there's a whole like family of Hermiticae or whatever. Like, that's impossible. There's no way that's possible. And I was like, so disappointed to find out that it's not true.
No, I just made that up. Sorry. I'm a Muppets fan. Kermit is actually my favorite character.
Muppets are great, but I'm also sad now, so
Oh,
thanks.
I'm sorry. I've taken away your innocence, Paul. I see. Anyway, yeah, there's no such thing as Comedicus chilius, but there are lots of wood frogs that freeze quite readily in winter, particularly in very northern and southern latitudes. If you think kind of boreal forests in Siberia and things like that, right, or in Alaska. So you do actually find frogs that look like this, and then if you stick them, like kind of turn up the thermostat a little bit, they will basically defrost and they'll come back to life. Now, what did you say, Vivian? Yeah, so there are lots of different approaches, right? And so you can really, and there's overlap between them, right? So it's not that it's just full on molecular for this or just behavioral or physiological for this group. There's a lot of overlap between the different two. It's very much like a smorgasbord in the sense of, you know, you pick from an array. Well, you don't pick, right? Because evolution doesn't work that way. But there's a whole bunch of different solutions to the same problem. Yeah, they do. They accumulate urea ahead of time as the temperature drops. They also increase glycerol and trehalose production. Where are those at? Yeah, so these chemicals here are called cryoprotectants. Actually, let's just jump around anyway because it's kind of more fun. It means I can answer Vivian's question better. They also produce things called antifreeze proteins, which are particularly cool. So if you look at, where's that picture gone? This picture here, you see these ice crystals that are kind of accumulating around the surface. It's fine on the outside, doesn't make any difference, but you really don't want those inside your cells because what they do, they will break open proteins and they will cause cells to lyse. If you've ever tried freezing strawberries, right, or peas, or I don't know, any kind of watery, high water content vegetable, and you do it slowly, what you'll tend to find is that when you defrost them, they'll be mush. Actually, strawberries are a great example. We just froze some. They're never going to have that kind of crisp texture, and the reason for that is because those ice crystals growing slowly kind of get bigger and bigger, and they kind of just like poke holes in things, so that when that strawberry is defrosted, all that water leaks out. There's no kind of cell structure to maintain its shape. And so avoiding ice crystal formation is actually one of like the prime objectives of psychrophiles, both the ones that hibernate or freeze and the ones that also live at low temperatures. And so one way of doing that is essentially to increase the osmolarity of your either circulating fluid or of your cytoplasm, and doing so basically drives excludes water, right? Obviously you have to means of it not drawing more water in, but you want to reduce the amount of water available for nucleation, right, for forming those ice crystals. One of the other things which is super cool, these things called antifreeze proteins or antifreeze glycoproteins. And, scientists kind of hate them being called that because they're not like, the stuff that you pour into your engine. They're not antifreeze in that sense. But what they do is they bind to ice crystals and they prevent nucleation, right? So they prevent ice crystals both growing and also recrystallizing. So what water there is in the cell or in the blood or whatever stays more fluid for longer at lower temperatures than it would without these, right? So essentially it doesn't, it's not like it lowers the freezing point, because it still wants to freeze, but it can't. So it kind of does lower the freezing point, but it doesn't lower it in the way that adding like salts and things like that would do so. So yeah, Vivian, it's exactly the same as what Dr. Lutnisky probably talked about. Where are we at? So those are cryoprotectants, and these are again, it's not, you can't delineate whether it's cold tolerant versus cold loving based on whether they use these or not, because you see these in all kinds of different organisms. But if you split it into various adaptations, right, you can really think about it in terms of molecular adaptations, right, which we'll talk about now. So these are very much similar to what we saw in barophiles, piezophiles, right? Essentially, cold constrains motion, same as pressure does. So the colder the environment, the less movement things have, right? And you see that if you look at that in solution, right? You know, basically as you reduce the thermal energy, then molecular motion decreases, right, over time until it ceases, absolute zero. And so in cyclophiles, in terms of protein and cell membrane structures, it's very much the same as the adaptations to high pressure, because pressure does the same thing. It constrains motion, right? And so the adaptations are very similar. And this is a beta-galactosidase protein. So this is something that turns lactose into glucose and galactose. And so it's a bacterial protein. And what they did was they searched, basically did a comparative bioinformatics kind of approach, looking at beta-galactosidases from cyclophiles versus mesophiles. And what they found was that there's a lot of changes which basically increased the flexibility of the protein, right? So increased the ability for that protein to move and maintain, well, essentially maintain its flexibility at lower temperatures, right? So it's very, very similar to the same approach to higher pressures. You'll also see the same with fatty acids. So in terms of both structural lipids, such as phospholipids, as well as storage lipids like TCA, tricarboxylic acid. It's got all the other different names for it. Basically what makes us fat. These up the top are what you would see in warm-blooded mesophiles like us, right? So organisms with an elevated internal temperature, you'll find a lot of saturated fatty acids, right? That's why bacon is supposed to be bad for you and fish oil is good for you, right? Essentially. Now, if you look at as temperature drops, you want to maintain membrane fluidity, and you do that by spacing those phospholipids further apart. Also, storage fats, you don't want them to solidify, right? You want them to be accessible to, whatever enzymatic reaction, be able to break them down or organize them or whatever. And so this is a monounsaturated fatty acid, right? And so you can see that cis bond here. This is that double bond, right? Carbon-carbon double bond. And because it's a cis bond, it actually kicks the fatty acid out, right? Kinks it at an angle, like 45 degrees or thereabouts. Now, if you have more than one of those, it gets all kinds of weird. So these are, hang on a sec. Hey fam, kiddos, hey Evie, don't forget I'm on the audio, okay? And they're talking about baking something. So you can see when they're polyunsaturated fatty acids, there's crazy shapes, right? They even curl all the way back around themselves like this. I'm not sure how that's pronounced. Anyway, and so that basically, along with other things like cholesterol, spaces out those phospholipids or even or TCAs and stops them compacting down and becoming immobile, right? And so that's, if you remember from the quiz, essentially if you take one of us, like, or a mesophile, and you reduce the temperature, that ordering becomes higher and that membrane will become waxy. It'll become more solid, right? And because a lot of things rely on membrane fluidity to work, such as transmembrane transport, cell signaling, all kinds of things, that will stop. So these are really, really important. And this is also why you get fish oil, right? It's not an accident that it's called oil. You don't get fish lard, right? Or fish butter, which would be kind of weird. So that's why fats from fish, which typically live at around 4 degrees Celsius, give or take, are fluid and oil-like at room temperature, whereas animal fats are solid, because room temperature is typically a lot lower than warm-blooded internal temperature. Proteins would fall apart. No, that's heat. That's an increase in thermal energy, Robertin. So if you think of a protein, there's going to be my poor attempt at trying to model this with my hands, right? So this will be, let's get it back here. Let's say this is a protein, like that. Actually, some proteins do that. And it's not just a joke. Essentially, each atom, right, in that is going to be vibrating a little bit, right? So it's just, if you look at any molecular animations, they all jiggle around a little bit, just because they do, right? But they stay within a certain range of motion, because otherwise they would lose their shape and not work, right? So if it's a Venus flytrap protein, for example, right, it's got to be able to kind of open and close like so, right? Now, if you put in more thermal energy, right, that vibration becomes higher, right? And then it's no longer able to do its job because it loses its shape, denatures, right? And so the forces constraining that motion are the bonds between the various R groups and secondary structures in that protein, right? So if you think my fingers are like alpha helices or something, right? They're kept together by bonds between the different R groups. Could be, ionic bonds, hydrogen bonds, covalent bonds, maybe, right? And so that constrains that motion. But the more energy you put into that particular structure, the more likely those bonds are going to be broken, right? So it could just go like that, right? And that structure is now longer not going to have the correct shape, and it's not going to work, right? So it's all really basic thermodynamics. Now, when you so we've got this kind of Venus flytrap protein here. When you reduce the thermal energy, right, that jittering becomes less and less, right? And so the space occupied by those atoms decreases, right? Because they're not zipping around against each other, right? And so as that space decreases, the movement reduces, the flexibility reduces, and now you've got something that can kind of do this, right? It's not able to move as much because it doesn't have the degree of flexibility and space that it would do normally, right? Because all of those atoms, they're basically coming closer to each other and their movement is becoming less. And so the structure becomes more constrained, right? And I'm not going to talk about water here because that's a terrible example. It behaves in completely weird ways, right? But and so essentially the adaptations, and it's the same for phospholipids in membrane. Here you go. So these are phospholipids, right? So, you know, typically they're kind of jiggling around like in our body, right? We're at 37 degrees, give or take. So there's a lot of thermal energy in there, right? So they're constantly like moving around like so. Now, if you take our fat out of us, right, and you stick in the fridge, right, which would be kind of gross. If anybody's read Fight Club, they'll get some flashbacks about this. And essentially what happens is that jiggling reduces and those phospholipids pack together more tightly, right? Now, if that's in a normal living cell, right, there's things that move around in that plane, in that phospholipid membrane, and things that go through it. There's kind of big proteins, right, that allow stuff to transport through them. And so if they're not able to move around or have the correct structure, you know, now let's say that channel is going to be shut, transmembrane transport isn't going to work, or different receptors aren't going to be able to come together and be activated. You know, so in psychrophiles, you don't have phospholipids like this. Oh, shit, I can't really do it very well. You have phospholipids like that, right? And so those phospholipids can't physically get as close to each other, right? Because these fatty acid tails, they're rigid, particularly when you get these double bonds, right? That kink, it's like bending a piece of a spring, right? We bend it into a new shape, it's going to retain that shape. It's not going to bend back, right? And so now when you have those phospholipids, right, even when they're a lot lower in temperature, right, they're not going to be able to get close to each other and pack down. So you're still going to be able to get things moving around, essentially, in that membrane, because the phospholipids aren't as physically close to each other. So that's really the idea. If you just think about it in terms of just Brownian motion, right? How do things move around and jiggle, 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 jiggle in response to temperature? More temperature, more jiggling, less temperature, less jiggling. And that's the whole idea behind absolute zero, which is what, 0 Kelvin, minus 273 Celsius. Though, I think that's right, something like that. Atoms have no motion at that temperature, right? It's they're a solid state. Those atoms don't move. It's a very, very hard state to get things into. Did that answer the question? So you can also see how temperature has the opposite effect too. You know, if you have those highly saturated, unsaturated, sorry, fatty acids, right, which in cold temperatures are able to kind of stay like so, and then you increase the temperature, right, they're just going to fall apart, right? So that's what defines in many ways not the only ways, but some ways, the temperature band in which organisms can live in, right? And psychrophiles are no different, right? It's not that they can live in like super, super warm places. They have their own band of temperatures in which they're capable of living at. Anyway, so just to round up, there's also actually something I didn't know before kind of looking into this again. But there's also an issue with RNA and DNA. I didn't actually know this. So the same applies, right? And this is really weird because I never thought about DNA as being affected by the cold. But DNA is more resistant to being unwound at low temperatures. So psychrophiles have a lot more helicases and isomerases, enzymes that kind of unwind DNA. And also they have what are called cold shock proteins, right? So when you cool down an organism, right? Obviously there's all these other things that happen. But also RNA forms secondary structures, right? Which you can see down here, right? RNA likes forming loops. right? Because it likes kind of, you know, like tRNA, for example, likes looping back on itself. And that's more likely to happen the lower the temperature, even if it doesn't normally do so at normal temperature for that organism. So if you cool down, say us, you know, down our cells down to 10 Celsius, other than lots of other bad things happening, also what will happen is mRNA forms secondary structures, and that blocks translation. And so obviously, not only do you have all the other issues of cold, such as protein function and membrane function and all that, you're not likely to produce the proteins you need to do whatever you need to do. And so cold shock proteins actually bind to those mRNAs and hold them in the correct conformation so that they can be translated. They're still slower, right, than before, but it works, right? And so that's something that I never knew about, which is super cool. Okay, and so one thing that you're probably thinking about is like, yeah, that's all like molecular stuff. That's not interesting. And it is interesting because it's how these things work. But there's also a bunch of kind of behavioral and physiological adaptations, which are super interesting, right? And a lot of these you already know about, even if you might not have thought about them in this way. So the classic one is leaves dropping in autumn. Now we kind of have that here in Texas, yeah, sort of. But essentially that's a cold response, right? So those trees basically stop growing during the cold. You can actually see that in their tree rings, right? That's why you get tree rings. And these large leaves with like the large surface area are very vulnerable to freezing. And so for deciduous trees, like maples, for example, or this thing that's an oak, I'm not 100% sure, but it's a better trade-off to just shed their leaves, right? Because they're not going to photosynthesize much because it's cold and not very sunny. And it will take a lot of effort to maintain those structures in adverse environments, so they lose them. Now, if you look at conifers, like evergreens, they take a very different approach. So these can actually photosynthesize even at cold, in winter in the cold. But what they have is they have a very like circular structure, needles, right? So they have a similar to these super cute chubby seals over here. They have a high volume to surface area or low surface area to volume ratio, right? So if you want to retain heat, you want as little surface area as you can for a given volume. And they're also very waxy, right? Pine needles are really hard to break down. And that's because they have a very waxy covering which protects them against cold. And also their structure, right? You know, conifers are this shape for a very specific reason, and that's to be able to shed snow. So if you look at all the snow that is around, this is boreal forest, I think, from Siberia. Hang on again, Kiara, I'll get back to you in a second. They're actually structured to be able to take on heavy snow loads, but then shed them. Whereas if you ever, you know, obviously it doesn't snow in Texas, but up in the Northeast, sometimes you'll get like fall snowstorms before the leaves have properly dropped, and there are tree limbs falling all over the place because they can't support that weight. Right. Anyway, okay, let's go back to Kiara. Oh, okay. So what bit don't you understand? The effect of cold on flexibility, or the adaptations to maintain flexibility?
Like the adaptations.
Oh, the adaptations. So typically the adaptations are more loops, right? So more hinges, right, to allow those parts of the protein to flex, right? And also reduced strength of bonds between the different parts of the protein. So it's actually easier to think about this in terms of heat. in a sense, because they're polar opposites, right? So you can, in terms of heat, you put a lot of energy in, you need stronger and stronger bonds to hold everything together to resist that heat, right? So hyperthermophiles, for example, have a lot of covalent bonds, cysteine disulfide bridges, right? And that's because there's more heat energy going in that wants to just make these things fall apart. The opposite is the case in psychrophiles. So you want less or weaker bonds holding the protein together so that there's less constraint on motion. Does that make sense?
Yeah, that makes sense.
Yeah. Yeah, so that's really the, sorry, dog's come back. That's really the idea behind it. There's also changes in hydrophobicity. So in psychrophiles, as with barophiles, the internal part of the protein is more hydrophobic, right? I'm not really sure what that does in terms of cold. And barophiles, it makes sense because it's all about water. But yeah, and also there's more of a preference for more flexible protein secondary structures, so like alpha helices versus beta pleated sheets in psychrophiles. So things which reduce the rigidity of the protein are favored at low temperatures. Oh, dog. Sorry, dog's back. What do you want? There you go. She's showing you a toy. She wants to play. You funny dog. Give here. Now she's just chewing and ripping the stuffing out of it. Yeah, anyway, so it's really about reducing the rigidity of the protein, because it's again that idea you want to maintain flexibility within a very narrow window, right? Because otherwise it's not going to have the correct either conformation to do its job or be able to move, right? Because proteins need to change shape to be able to do their job typically. And so maintaining that flexibility is key, right? So if you reduce the energy in the system, then you need less constraints on that motion to maintain flexibility, and vice versa. So really that's how it works. Did that help? Cool. Okay. So in terms of physiology, so behavior adaptations we kind of covered with the bears, right? But there are also other bears in the Arctic. So this is a woolly bear moth caterpillar, super cute, much smaller than actual bears. But this is particularly interesting because it lives quite happily in the Arctic, up in northern Greenland. And it basically takes about 7 years to turn into a moth. And the reason is that it spends the vast majority of its time frozen. And so when things get roasty toasty in the Arctic, when they get up to like 10 degrees Celsius, this thing thaws out and wanders around eating stuff. And then once it's had a dinner, it will find somewhere sunny to bask. So it basically lies, well, not lies, because, it's a caterpillar, stays in the sun so that the sun, it gets heated up. And so its digestive reactions, right, its biochemical reactions increase. And so it's actually a very efficient digester of things because it uses the sun to warm itself up to speed up those reactions. And it survives down to like minus 45, 50 degrees Celsius typically. And then, does a little bit more of its life cycle one summer, and then it'll freeze again, and then it'll do a little bit more next summer, and so on and so forth, until it's actually able to turn into a moth and fly around and reproduce. So there's a lot of that, as with other extreme environments, matching growth and reproduction to the conditions that are favorable for that, and then just stopping everything, put into stasis until that comes back again. You see that in deserts a lot. You know, there's very little life until it rains, and then it all just goes crazy. You know, frogs kind of come out of the sand and all kinds of things. And then there's also a lot of other physiological adaptations, which we don't really have time to go into, such as countercurrent exchange, which is how penguins stand on ice without getting cold feet. Well, they do get cold feet. That's kind of the point. But also one of the big ones is this whole thing of fat and round. So if you want to conserve heat, you want to have the smallest amount of surface for your volume. And the funny thing is that applies also to hot environments. So elephants are so long-lived because they have a very small surface area to volume ratio, right? So they're warm-blooded. They have to actively maintain an elevated temperature, but they need a lot less energy to do so than mice do, for example, which have a very high surface to volume ratio. And so whenever you look at particularly mammals, right, which have to maintain an elevated body temperature, they will always be fat and round, right? Any mammal that lives in a cold environment will have a large volume to its surface area. They do that both by having lots of fur. Arctic foxes, for example, have really, really small ears, like tiny little nubbins. If you compare that to a desert fox, for example, which have these huge ears, is another example of that. And also these, I think they're king penguins, or they might be emperor penguins, essentially doing the same thing down here as they're overwintering on the Antarctic ice shelf. You know, essentially this is reducing the surface area of the individuals on average, right? And they shuffle around and they rotate, so they're not always on the outside the whole time. So essentially it's all about conserving. You can do lots of different things. You can just not worry about the temperature and just freeze, right? A lot harder for mammals than it is for insects. You can change your structure and your shape to survive, or you can shut down non-essential functions. Or if you do need to keep living, then there's a lot of other things that you can do to adapt to those environments. And if anyone's seen March of the Penguins with Morgan Freeman, it's really well worth watching. It's always great just listening to his voice anyway. So that's pretty much it from me in terms of stuff for this. This is also our last kind of biology part of astrobiology, because on Friday we're going to be starting on exoplanets. You know, what they are, what they look like, discovery of, things like that. So this is kind of the end of the biology part of astrobiology. Does anybody have any questions? Thoughts, ideas? Well, if you do, bring them with you on Friday and we'll go over any questions you have there. Okay, cool beans, everybody. Thanks for your time, and we shall see you on Friday.
Have a good day.
You too.